Hello, welcome you all to the biology classes, and we are here in the 12th lecture of our biomolecules, right? In the last class we were discussing about the enzymes. Okay, today before going to the detailed explanation about the enzyme and the enzyme action, we will discuss briefly about the reactions, so that we will get clearly idea about the action of enzymes on a particular reaction, action of the enzyme on a particular reaction. Okay, so when we say the word reaction, there must be something that should become some other thing, means there must be some two things which will combine together, will react together, or they will form some other products. But the reaction may be physical reaction or the reaction may be chemical reaction. Of course, it happens in nature or in living systems. Okay. Physical reaction means just the change in the shape. Okay, just the change in the shape without any breaking of the bond or no formation of bond. Okay, just it makes the change in the shape of the molecule, physical change or visible change. But in case of chemical reaction, some bonds will be broken down, some new bonds will be formed. Maybe any kind of reaction. Okay, maybe inorganic reaction or organic reaction. But in both of them, the bonds are broken down. Some bonds are broken down and some bonds are formed. Okay, so when we look at inorganic reaction, again the inorganic chemicals are involved. When we look at the organic reaction, the biomolecules are involved, like proteins, carbohydrate, nucleic acids, lipids, vitamins, etc. All those biomolecules are involved. Okay, but here for these organic reactions, we need the help of the enzymes. Organic reactions, we need the help of our enzymes or biocatalysts. Okay, we will see some example for this inorganic reaction as well as organic reaction. So when we take a barium hydroxide, when it combines with a sulfuric acid, so it will form barium sulfate. And the remaining thing will be two molecules of water. Okay, just here one compound is broken down, this compound is broken down. So here the barium and the sulfate combined together will form a barium sulfate and one water molecule will be released. Okay, but when we look at the example for organic reaction, maybe the starch, for example, the starch will form glucose. Remember, it is a polysaccharide, it is a monosaccharide. Okay, so for the breakdown of the starch, the enzyme is amylase. Okay, the process is known as hydrolysis. Means the process of breakdown of starch into glucose by the action of the enzyme amylase is known as hydrolysis. So this is an example for a inorganic reaction or here the organic reaction. Okay, so we don't need enzyme in this reaction, but compulsorily it must have an enzyme for the reaction to occur, then only the process will be completed. So here we have amylase enzyme. This is the enzyme that will speed up the breakdown of starch into glucose, polysaccharide into monosaccharide. Right. Here one more thing we can imagine that every reaction has a particular speed and particular amount of product is formed. In every reaction a product is formed. This is the product. In this reaction, this is the product. This is the product. Okay. Here, when we count the time and the product, means in a particular time, how much amount of product is formed. That is known as a rate of a reaction. That is known as a rate of a reaction. Okay. So here, rate of reaction means amount of product formed per unit time. Amount of product formed per unit time. ಅಂದರೆ ಒಂದು ನಿಮಿಷಕ್ಕೆ ಎಷ್ಟು ಪ್ರಾಡಕ್ಟ್ ಫಾರ್ಮೇಷನ್ ಆಯಿತು, ಒಂದು 10 ನಿಮಿಷಕ್ಕೆ ಎಷ್ಟು ಗ್ಲೂಕೋಸ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ಗಳು ತಯಾರಾದವು. ಲೈಕ್ ದ್ಯಾಟ್ when we consider the time limit as a criteria for counting the rate, the product that will both time as well as the product amount together will form the rate of a reaction. We can express this rate of reaction in the form of rate equals to delta P divided by delta T, amount of product per unit time. Okay, remember this equation. It may be helpful for your examination. Exam help ಆಗ್ಬೋದು equation ನೆನಪಿರಲಿ. ಸಿಂಪ್ಲಿ ರೇಟ್ ಆಫ್ ರಿಯಾಕ್ಷನ್ ಅಂದ್ರೇನು, ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಟೈಮ್ನಲ್ಲಿ ಎಷ್ಟು ಪ್ರಾಡಕ್ಟ್ ಫಾರ್ಮೇಷನ್ ಆಗಿದೆ, ಅಮೌಂಟ್ ಆಫ್ ಪ್ರಾಡಕ್ಟ್ ಫಾರ್ಮ್ಡ್ ಪರ್ ಯೂನಿಟ್ ಟೈಮ್, ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಪ್ರಾಡಕ್ಟ್. ಓಕೆ, ರೈಟ್. ಹಿಯರ್ in rate of reaction, the direction is not mentioned. The reaction may be forward, the reaction may be backward. Reaction may be forward or the reaction may be backward. The direction is not known. But when direction is known, the rate is called a velocity of reaction or speed of reaction. Okay, so we can write here, when direction is known, direction of reaction is known, then rate of reaction is called velocity or simply speed. I'm telling you, don't go for the physics formula derivation, nothing that. Just simply, when reaction is not known, it is called, when direction is not known, it is called rate of reaction. When direction is known, it is called velocity of reaction or speed of reaction. That's it. Okay, don't make yourself complex. Again, don't think in the physics manner, right? Just direction is not known, it is called rate of reaction. Direction is known, it is called velocity of reaction or speed of a reaction, right? That's it. Don't make yourself complex by connecting these things to the physics, right? I'm telling you again, pause the video, take a notes in your book. Okay, so here when we say the rate of reaction, this will be affected by many factors. We have a temperature, we have a light intensity, we have a water, we have some other chemicals called inhibitors. Like that, rate of reaction is influenced, affected by many factors. Influenced or affected by many factors. Okay, so one of such factors is the temperature. One of such factors is the temperature. Okay, better I'll write the part next. So rate of reaction is affected by temperature. Rate of reaction is affected by the temperature. So on that base, there is a rule called rule of thumb. Rule of thumb. Thumb is a name of scientist. Okay, don't ask what is that scientist, don't need of at this age now, just rule of thumb. So according to this rule of thumb, the rate of reaction doubles or reduces by half per every 10 degrees Celsius change in either direction. Okay, rate of reaction will double or reduce to half. ಅಂದರೆ ಸ್ಪೀಡ್ ಡಬಲ್ ಆಗ್ಬೋದು ಅಥವಾ ಅದರಲ್ಲಿ ಅರ್ಧ ಆಗ್ಬೋದು. ಎಷ್ಟು ಸ್ಪೀಡಿಂದ ಹೋಗ್ತಾ ಇದೆ ಆ ಸ್ಪೀಡ್ ಡಬಲ್ ಆಗ್ಬೋದು ಅಥವಾ ಆ ಸ್ಪೀಡ್ ಅದರ ಅರ್ಧ ಆಗ್ಬೋದು. ಯಾವಾಗ? ಪರ್ ಎವ್ರಿ 10 degrees Celsius change in the temperature in both directions. Either the reaction may be forward reaction or the reaction may be backward, backward. But when 10 degrees Celsius increased, reaction may be increased by 2 times. When the temperature is reduced by 10 degrees Celsius, 50 to 40, the reaction may be reduced to half. The speed may be reduced to half. Means half of that speed. That is called as rule of thumb. Remember, it's for 1 mark. It may be helpful for you. Okay. So rule of thumb, rate of reaction is affected by the temperature. That is by 10 degrees Celsius change in either direction. It may be doubled in forward direction or reduced to half in backward direction. Okay, that's about the effect of temperature on the rate of reaction. Okay, so we have one more equation, one more factor called catalyst. Main factor is temperature. One more factor is catalyst, enzyme. Okay, so uncatalyzed reaction, the rate is slow. Then catalyzed reaction, here the rate is faster or more. ಅಂದರೆ ಎಂಜೈಮ್ ಇದ್ದರೆ ರಿಯಾಕ್ಷನ್ ಫಾಸ್ಟ್ ಆಗುತ್ತೆ, ಎಂಜೈಮ್ ಇಲ್ಲ ಅಂತಂದರೆ ರಿಯಾಕ್ಷನ್ ಸ್ಲೋ ಆಗುತ್ತೆ. ವಿತ್ ದ ಎಂಜೈಮ್ ದ ರೇಟ್ ಈಸ್ ಮೋರ್, ವಿದೌಟ್ ದ ಎಂಜೈಮ್ ರೇಟ್ ಈಸ್ ವೆರಿ ಲೆಸ್. ವಿಲ್ ಟೇಕ್ ಅನ್ ಎಕ್ಸಾಂಪಲ್. ಸೊ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, we have a CO2 plus water molecule. Both will combine together to form a compound called carbonic acid, H2CO3. It is carbonic acid. Right. Without the enzyme also the reaction will continue. But in this speed, without the enzyme, there are about 200 molecules of carbonic acid per hour. ಅಂದರೆ these many molecules of carbonic acid are formed in 1 hour without enzyme. Only 200 molecules of carbonic acid are formed. But when there is an enzyme called carbonic anhydrase, the speed will be increased by million times. Means the reaction speed will be increased by million times. For example, about 6 lakh molecules of carbonic acid are formed in a second. Okay, 6 lakh molecules of carbonic acid are formed in just 1 second. So look at the speed of the reaction with the enzyme. Whenever there is no enzyme, only about 200 molecules of carbonic acid are formed in 1 hour. But when the enzyme is present, about 6 lakh molecules of carbonic acid are formed in 1 second just. Okay, imagine the speed of the reaction with the help of an enzyme. Okay, so when we have many reactions in a chain, in a series, what we discussed in the previous class, this is what we called as a metabolic pathway. Metabolic pathway. Okay, for example, we have a glycolysis. Glycolysis, one glucose molecule by many reactions will form 2 molecules of pyruvic acid. Okay, we will discuss in chapter 14 in respiration in plants. Okay, here whenever the oxygen is present, the process is called as a glycolysis. It will name it glycolysis. Whenever oxygen is present, okay, when oxygen is not present, mainly in our muscle cells, when oxygen is not present, this will form lactic acid. O2 is absent. When oxygen is absent, mainly in our muscle cells, the glucose will form lactic acid. Okay, in the muscles, if oxygen is present, glycolysis will form 2 pyruvic acid molecules. When oxygen is absent, this will form lactic acid. Okay, again we will discuss that in our respiration chapter number 14. Okay, don't worry about that. Now the main important concept of today's class is nature of enzyme action. Okay, the main theme of this enzyme concept, that is nature of enzyme action, mode of enzyme action, or mechanism of enzyme action, we will discuss briefly in this class. Nature of enzyme action. Okay, in many books it is referred as mode of enzyme action, and somewhere it is referred as mechanism of enzyme action, etc. Everything is in. Andre enzyme action yav ರೀತಿ ಆಗುತ್ತೆ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. Let us see what is that. Okay, ನೀನೆ ಕ್ಲಾಸಲ್ಲಿ ಹೇಳಿದ್ದೆ ಲಾಸ್ಟಿಗೆ ಏನು, enzyme will bind with the substrate, and this will form a enzyme substrate complex. Later on that will convert into enzyme product complex. Later on this will release the enzyme and product. Okay, so better we make different colors. Enzyme plus substrate will form enzyme substrate complex, that will form enzyme product complex. We will get a enzyme plus product at the end. Okay, so enzyme plus substrate will form enzyme substrate complex. Then this converts into enzyme product complex. Lastly, this will be separated each other. So enzyme gets separated and product gets separated. Okay, so this is one catalytic cycle. One catalytic cycle. One catalytic means one cycle of the enzyme action. ಅಂದರೆ ಒಂದು ಎಂಜೈಮ್ ರಿಯಾಕ್ಷನ್ ಸ್ಟಾರ್ಟ್ ಮಾಡಿ ರಿಯಾಕ್ಷನ್ ಯಾವ ರೀತಿ ಎಂಡ್ ಆಗುತ್ತೆ. ಓಕೆ, ಮೊದಲೇನು ಮಾಡ್ತದೆ, ಎಂಜೈಮ್ ಸಬ್ಸ್ಟ್ರೇಟಿಗೆ ಬೈಂಡ್ ಆಗ್ತದೆ, ಆಮೇಲೆ ಏನಾಗ್ತದೆ, ಎರಡು ಕಂಪ್ಲೀಟ್ಲಿ ಬೈಂಡ್ ಆಗ್ತವೆ. ಆವಾಗ ಏನಾಗುತ್ತೆ, ಇಟ್ಸ್ ಕಾಲ್ಡ್ ಎಂಜೈಮ್ ಸಬ್ಸ್ಟ್ರೇಟ್ ಕಾಂಪ್ಲೆಕ್ಸ್. ದೆನ್ this enzyme substrate complex will form a enzyme product complex. Enzyme product complex. Later on, this will be completely get separated. That's called enzyme plus product. Again, this enzyme is ready for the next reaction, because this is end of one cycle. Enzyme was free here, enzyme is again free now here. Again, the enzyme can bind with another substrate. Here we can label them S1. We can take this as a S2. I mean substrate 1, the first reaction. This is substrate 2, the second reaction. Again, that will form ES2 complex. They will form EP2 complex. We'll mention EP1, EP2. Okay, so EP2 complex. Then they will form enzyme plus product complex. Again, the enzyme is ready for next reaction. Again, the enzyme is ready for next reaction. ಅಂದರೆ ಒಂದು ರಿಯಾಕ್ಷನಲ್ಲಿ ಏನು ಮಾಡ್ತು, ಎಂಜೈಮು ಒಂದು ಸಬ್ಸ್ಟ್ರೇಟ್ನ ಒಟ್ಟಿಗೆ ಬೈಂಡ್ ಆಗಿ ಅದನ್ನೇನು ಮಾಡ್ತು, ಒಂದು ಪ್ರಾಡಕ್ಟ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಮಾಡ್ತು. ಅದನ್ನೊಂದು ಪ್ರಾಡಕ್ಟ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಮಾಡ್ತು. ರೈಟ್. ಸೊ ಒನ್ ಸಬ್ಸ್ಟ್ರೇಟ್ ಹ್ಯಾಸ್ ಬಿಕಮ್ ಅ ಪ್ರಾಡಕ್ಟ್. ಮತ್ತೇನು ಮಾಡ್ತೀವಿ, ಎಂಜೈಮು ಇನ್ನೊಂದು ಸಬ್ಸ್ಟ್ರೇಟ್ನ ಒಟ್ಟಿಗೆ ಬೈಂಡ್ ಆಗ್ತದೆ, ಸೆಕೆಂಡ್ ಸಬ್ಸ್ಟ್ರೇಟ್. ಅಗೇನ್ ಇಟ್ ಕನ್ವರ್ಟ್ಸ್ the another substrate into another product. Like that, this enzyme will continue, keep on changing the substrates into the various products. This is one catalytic cycle. Okay, bookal 4 steps ಕೊಟ್ಟಿದ್ದಾರೆ, ಅದನ್ನ ನೋಡಿ ಬರ್ಕೊಳ್ಳ್ರಿ. Nothing, just first step they will bind together. Second step they will diffuse together each other, ಅಂದರೆ ಎರಡು ಕಂಪ್ಲೀಟ್ಲಿ ಕೆಮಿಕಲಿ ಬೈಂಡ್ ಆಗ್ತವೆ. Then what happens at this reaction, the structure of the substrate becomes structure of product. ಅಂದರೆ ಇಲ್ಲಿ ಸಬ್ಸ್ಟ್ರೇಟ್ನ ಸ್ಟ್ರಕ್ಚರ್ ಯಾವ ರೀತಿ ಇತ್ತು, ಅದರಿಂದ ನಮಗೆ ಯಾವ ರೀತಿ ಪ್ರಾಡಕ್ಟ್ ಆಗಬೇಕಾಗಿದೆ ಅದರ ಸ್ಟ್ರಕ್ಚರಲ್ಲಿ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ಈ ರಿಯಾಕ್ಷನಲ್ಲಿ ಸಬ್ಸ್ಟ್ರೇಟ್ ಯಾವ ರೀತಿ ಇತ್ತು, ಅದರಿಂದ ನಮಗೆ ಯಾವ ರೀತಿ ಸಬ್ಸ್ಟ್ರೇಟ್ ಪ್ರಾಡಕ್ಟ್ ಆಗಬೇಕಾಗಿದೆ, ಆ ಪ್ರಾಡಕ್ಟ್ನ ರೀತಿ ಸ್ಟ್ರಕ್ಚರ್ ಮಾಡಿಫೈ ಆಗುತ್ತೆ. ಓಕೆ, ಒನ್ ಮೋರ್ ಥಿಂಗ್ ಯು ಶುಡ್ ರಿಮೆಂಬರ್ ಹಿಯರ್, at this level, mainly in this level, the substrate to convert into product, there is a lot of change in the energy. Okay, so means the structure keep on changing, lastly to form a product. One better we have it as a enzyme substrate complex, and we have a enzyme product complex. So to convert the enzyme substrate complex into enzyme product complex, this complex has to undergo many structural changes, and at some point it will reach high energy state. This high energy state is called as a transition state. This high energy state is known as a transition state. It means this is the point where the structure of the substrate converts into structure of the product. Okay, this is the point. Okay, should remember, from enzyme substrate complex, it's going to form enzyme product complex. In that process, this complex will reach at high energy state, maybe the temperature, maybe pressure, maybe the concentration of the product substrate, anything that will keep on increasing. At this point, it is the highest stable state. It is a more stable transition state. It means more stable. Stable means energy content of the molecules. Okay, these molecules have a high energy now, so it is considered as more stable or a transition state or high energy state. Okay, so among all these altered structural states, means it is going to change the structures to form a product. Among that, this is the high energy state. This is the high energy state. Okay, so better we'll draw a diagram to show this properly. Okay, we'll understand better. Okay, but you should remember that one enzyme can catalyze many reactions. One enzyme finish one reaction, ready for the next reaction, finish the second reaction, ready for the third reaction. Like that, one enzyme can take place, can undergo many catalytic cycle. Okay, we'll show this with the help of a diagram. So let us draw a small graph. Y-axis, x-axis. So this is x-axis, this is y-axis. So on x-axis we will take a progress of reaction. On y-axis, let us take a potential energy. On x-axis, y-axis potential energy, and x-axis progress of a reaction. Okay, so this is the energy. Take any unit, don't need to worry about the unit. Energy measured by any units. Okay, so this is the speed of reaction or a progress of how much reaction has happened. Okay, imagine this is the substrate. Better we'll make a dot. Means the substrate has this much of energy. Substrate has this much of energy. Okay, better I'll write that once again. Before that, it's easy to understand. So when we say the energy level or the potential energy, level. So when the energy level of the substrate is higher than energy level of the product, then the reaction is exothermic reaction. When the energy level of the substrate is lesser than energy level of the product, then the reaction would be endothermic reaction. The reaction would be endothermic reaction. ಅಂದರೆ ಈಗ ನಮ್ಮ ಹತ್ರ this is substrate will label as a S. This is our product. Okay, substrate has more energy, product has less energy. Okay, so substrate more, product less. The reaction is exothermic. Means exo release, thermo heat is released. But when the substrate has less energy, product has more energy, then the reaction is endothermic. Means heat is absorbed. Okay, in this case heat is released, in this case heat is absorbed. Right, here what happens, even though the reaction is exothermic, endothermic, spontaneous, whatever may be the type of reaction, the substrate must have to reach the transition state. I have shown you here earlier the substrate enzyme product. It has to reach the transition state, means high energy state or the stable state. Okay, so it has to reach the stable state, again come back to form the product. Come back to form the product. Okay, so imagine for this reaction enzyme is not there. Enzyme is not there. Okay, so the substrate needs more energy. Substrate needs more means it will reach the very high energy level. Reach the very high energy level. Okay, so this is the product level. Okay, here we can see this is the transition state or high energy state. Transition state or high energy state. But whenever the enzyme is present, whenever the enzyme is present, the substrate needs less energy. The substrate requires less energy. So it will reach a very less energy level and form the product. Form the product. This is also transition state. This is also the transition state. Transition state. Okay. Here we can find this is energy level, this is high energy level, this is also high energy level. Okay. So we can see this gap. See this gap. This difference, this gap is known as activation energy. Activation energy means earlier how much energy is present in the substrate, how much energy is required to form the product. This gap is called activation energy. This is also how much energy is present in the substrate, how much energy is required to form the product. Again this gap is also called activation energy. Better we will write the definition of the activation energy since it is important for the examination. So difference between the energy content of the substrate and that of the transition state is known as activation energy. Activation energy ಅಂದರೆ ಸಬ್ಸ್ಟ್ರೇಟ್ ಹತ್ರ ಎಷ್ಟು ಎನರ್ಜಿ ಇತ್ತು ಮೊದಲಿಗೆ ರಿಯಾಕ್ಷನ್ ಆಗೋಕ್ಕಿಂತ ಮುಂಚೆ ಪ್ಲಸ್ ಆ ಟ್ರಾನ್ಸಿಷನ್ ಸ್ಟೇಟಿಗೆ ರೀಚ್ ಆಗಲಿಕ್ಕೆ ಎಷ್ಟು ಎನರ್ಜಿ ಬೇಕಾಗತ್ತೆ. ಓಕೆ, ಸೊ ದಿಸ್ ಇಸ್ ಆಲ್ಸೋ ಒನ್ ಲೆವೆಲ್ ಆಫ್ ಎನರ್ಜಿ, ದಿಸ್ ಇಸ್ ಆಲ್ಸೋ ಒನ್ ಲೆವೆಲ್ ಆಫ್ ಎನರ್ಜಿ. This gap is known as activation energy. Okay, so practice the definition properly. Don't try to add any other words here. This much is enough for examination, 1 mark. Okay, so here this is the activation energy without enzyme. Here this is the activation energy with enzyme. ಓಕೆ, ಸಿಂಪ್ಲಿ ಕಾಂಪ್ಲೆಕ್ಸ್ ಮಾಡ್ಕೋಬೇಡ್ರಿ, ಕನ್ಫ್ಯೂಸ್ ಮಾಡ್ಕೋಬೇಡ್ರಿ. ಏನಂತಂದರೆ ಬೆಟರ್ ವಿಲ್ ಟೇಕ್ ದಿಸ್ ಲೈನ್ ಅವೇ ಫ್ರಮ್ ದಿಸ್ ಲಾಂಗರ್ ಲೈನ್. ಯು ಕ್ಯಾನ್ ಮೇಕ್ ಬೆಟರ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್ ಓವರ್ ಹಿಯರ್. ಓಕೆ, ಸೊ ನಮ್ಮ ಹತ್ರ ಸಬ್ಸ್ಟ್ರೇಟ್ ಹತ್ರ ಇಷ್ಟು ಎನರ್ಜಿ ಇತ್ತಪ್ಪ. for example, we'll make some units 1 2 3 4 5 6 7 ಎಷ್ಟು ಎನರ್ಜಿ ಇತ್ತು. ಓಕೆ, ಸೊ ಸಬ್ಸ್ಟ್ರೇಟ್ ಹತ್ರ ಎನರ್ಜಿ ಎಷ್ಟಿದೆ ಹೌದಾ, ಆದರೆ ಪ್ರಾಡಕ್ಟ್ ಫಾರ್ಮೇಷನ್ ಮಾಡಲಿಕ್ಕೆ ಅದು ಈ ಹೈ ಎನರ್ಜಿ ಲೆವೆಲ್ಗೆ ಹೋಗಿ ಬರಲೇಬೇಕು ಹೌದಾ, ಹೈ ಎನರ್ಜಿ ಲೆವೆಲ್ಗೆ ಹೋಗಿ ಬರಲೇಬೇಕು ಹೌದಾ. ಆದರೆ ಎಂಜೈಮ್ ಇದ್ದಾಗ ಸ್ವಲ್ಪ ಲೆವೆಲಿಗೆ ಹೋದರೆ ಸಾಕು ಪ್ರಾಡಕ್ಟ್ ಫಾರ್ಮೇಷನ್ ಮಾಡಬಹುದು. ಆದರೆ ಎಂಜೈಮ್ ಇಲ್ಲದೇ ಇರುವಾಗ ಅದು ಸಬ್ಸ್ಟ್ರೇಟ್ ಬಹಳ ಹೈ ಎನರ್ಜಿ ಲೆವೆಲ್ಗೆ ಹೋಗಬೇಕಾಗತ್ತೆ. ಮೀನ್ಸ್ ವೆನ್ ಎಂಜೈಮ್ ಈಸ್ ಪ್ರೆಸೆಂಟ್, the substrate requires less energy to form a product, but when enzyme is absent, the substrate requires more energy to form the product. So you should differentiate that. There is more activation energy when enzyme is absent. There is less activation energy when enzyme is present. ಓಕೆ, ಅಷ್ಟೇ. ನಿಮಗೆ ಕಾಮನ್ ಎಕ್ಸಾಂಪಲ್ ಕೊಡಬೇಕಂದ್ರೆ, ಒಂದು ರಿಯಾಕ್ಷನ್ ಆಗಲಿಕ್ಕೆ ಎಂಜೈಮ್ ಇದ್ದರೆ ರಿಯಾಕ್ಷನ್ ಬೇಗ ಆಗುತ್ತೆ, ಎಂಜೈಮ್ ಇಲ್ಲ ಅಂತಂದರೆ ರಿಯಾಕ್ಷನ್ ತುಂಬ ಸ್ಲೋ ಆಗುತ್ತೆ. ಓಕೆ, ಸೊ ಅದಕ್ಕೆ ಹೇಳೋದು ಎಂಜೈಮ್ ಈಸ್ ಮಸ್ಟ್ ಫಾರ್ ಎವ್ರಿ ಬಯೋಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್, ಫಾರ್ ಎವ್ರಿ ಬಯೋಮಾಲಿಕ್ಯುಲರ್ ರಿಯಾಕ್ಷನ್, or for every organic reaction in the plant body, in the animal body, in the body of any microbe. So enzyme is a must for every reaction. Okay, so practice this graph also, helpful for your examination. It may come in the form of 5-mark questions. Right, this is all about your nature of enzyme action. Okay, so in our text they have given like how do enzymes carry out high level of energy, something they have given. Okay, all about enzyme action only. It's all about enzyme action only. Okay, don't worry much about that. Okay, it's almost about the enzyme action itself. Right, so practice this graph, important for 5 marks. Right, so in the next class, maybe the last class of the biomolecules, we will discuss the factors affecting enzyme action. We will discuss the factors affecting enzyme activity. Okay, so I'll just mention the factors in today's class. I'll explain in tomorrow's class. Okay, so firstly, first factor, we have temperature will affect the enzyme activity. We have pH also affect the enzyme activity. We have the water which affect the enzyme activity, or which we have light intensity which also affect the light intensity. Next, the factors like concentration, substrate also affect the enzyme activity. Next, concentration of enzyme also affect the activity or speed of the rate. Lastly, we have a presence or absence of inhibitors affect the enzyme activity. Okay, so temperature, pH, water, light, concentration of the substrate, concentration of the enzyme, presence or absence of a inhibitor also affect the enzyme activity. This we will discuss in tomorrow's class. Alright, so draw this diagram in your book, don't send in the group. So for today there is no homework for you, right? So even then, if you have any more doubts, please forward the doubts towards the group. We'll try to come back to you with the answers very soon. Till that, keep learning. See you in the tomorrow's class. Thank you.